James, or Jimmy as we all know him, completed his BS in biology with minors in chemistry and psychology here at Salem State. He went on to complete a master's at the School of Natural Sciences, Forestry and Agriculture at the University of Maine, Orono. His thesis, using moose and ticks, was titled A Socio-Ecological Approach to Wildlife Disease Risk. Jimmy changed direction, embarking on a career in the biotech pharma field, starting at Enzymatics QIAGEN, before moving on to New England Biolabs in Ipswich, Mass. Here he is a process development scientist, and his role includes, and I quote from his CV, "My role extends to evaluating the potential of novel expression systems for recombinant protein manufacturing, pioneering advancements in clone development, and integrating cutting-edge ssDNA aptamer technology to achieve unrealized levels of impurity clearance." I have known Jimmy for over 10 years, and what stands out in my memory of him is his innate curiosity and willingness to try new things, surely the characteristics that any good scientist should possess. His talk today is entitled Of Moose and Men, Evolutionary Threats in Wildlife Disease and Vaccine Manufacturing. Welcome back, Jimmy.
Thank you for the kind introduction. It's surreal to be back at doing this. My high school teacher and his students attended today. I did not know that was going to be happening. Seeing a lot of the professors that taught me so much and the teacher that taught me so much 15 and 10 years ago, and doing this, something I came to every year with a lot of enthusiasm. It is a very special moment, so thank you so much for inviting me. This is the wildest title slide you probably ever see. This tries to tie in Darwin. This tries to tie in recapitulate my journey as an alumni from my time at Salem State here all the way till now where I'm at New England Biolabs. The work that I did at University of Maine involves moose. I was asked to tie that in a two-part talk, also talking about what I do at New England Biolabs. I wanted to relate this, the only thing that unifies the two topics of today, other than me and the silly title, is evolution. It unifies a little bit of everything, and I'm going to throw out there that this is really looking at natural versus artificial selection. If you hate that, just ignore it. About me, I started at Salem State in 2012. I had a great time here, started my journey in independent research with Aziz Almadi in my General Chemistry 1 class in my first semester in the lab. He gave me the opportunity to do water quality testing in Morocco through a lot of orphanages that were down there, and I had a great time, and it was the first time that I really got addicted to discovery. There was a jolt of energy that you could get when you found something that no one else knew. It was something that wasn't in a textbook, and that became a little bit of a drug for me. I also met my girlfriend at the time, my wife, Katie. I also got the opportunity to work at Cat Cove Marine Lab for about 3 years, and this was also working with the Division of Marine Fisheries. I was a part of the Boston Harbor Softshell Clam Enhancement Project, and got to work with a lot of very interesting people in a good way. And from there, explored more independent research, had an opportunity to spend a summer as a research assistant down in Woods Hole, working with Dr. Hans Laffer there for a summer. We investigated the effects on lobster development from treating larval and juvenile lobsters with various alkyl phenols. Think BPA, think things that we're concerned about when we talk about plastics. And that was very interesting. I also started working with Alan Young on green crabs. When I graduated, I kept working on green crabs because something was addictive about this little beast. And since then, Dr. Alan Young and I have worked on 3 papers together about various topics. When I graduated, I worked through the Princeton Review while looking for a job, ended up at Enzymatics QIAGEN, as Ryan stated. This is where I got my first feel for operations in biotech. I was kind of addicted to it. Ended up feeling like I was settling, as I didn't find any graduate position that worked out for all parties. Left there after getting a sort of survey of all different aspects of operations in the biotech industry for University of Maine. When I got there, we also collaborated, me and a few other people, as a co-founder of a greencrab.org, a nonprofit that is really focused on studying green crab as it relates to using them as a culinary product. Eat the enemy type of thing. When I was at UMaine, I worked on moose, hence the topic of today. Graduated with a master's, left UMaine early to have a job at New England Biolabs. Since then, you can see this starts to taper off. The amount of things that I have going on in my life. My daughter was born in 2022, took a board position for a short time at the Salem Sound Coast Watch, and then my son Levi was just born last September, and he's awesome. Today, I'm going to focus on, I'd rather just talk about my kids for 50 minutes, but I can't. So what we're going to talk about are these two things in the orange box. Moose and the work that I do at New England Biolabs, and it's going to be sort of split. So the first thing is the diversity of evolution of Anaplasma bacteria and the infections that it has in North American moose. When I got to University of Maine, I actually was accepted in a position with it would end up being my advisor, without a project. She said, you can work whatever you want, which if you know me, it's kind of dangerous. I got real excited about that and was a little overwhelmed with the possibility. There were a lot of new faculty who were trying to get into new projects. And if you can believe it, and up there, Orono was real up there in Maine, there was not a single person working on moose. What do you do when you go to Maine? You go up there, you see loons and you see moose, and that's just what you do. If you are associated with anything in Maine, the logo, the mascot, it always seems to be a moose. How are we not working on moose? It's very dense in Maine, the moose population, and there's a variety of subspecies of moose that are going to be all Alces, and what comes after it is the differentiator. What we're dealing with around here is the eastern moose, that's Alces americana, but the project has expanded since I started it back in 2017. To get involved with this, I reached out to the Inland Fisheries and Wildlife, the moose biologist. He said that he wasn't going to be working on any moose with UMaine students because all of the students that he takes is from the University of New Hampshire. So I drove all the way down to the University of New Hampshire, which is like 3 hours from Orono, scheduled an appointment with his old advisor, the person he was referring to, and said, I want to do work with moose, and here are my ideas. What I had talked to about that person is that when I was looking into ideas of how I wanted to spend my time in graduate school, I was really fascinated by entomology. It was a really great course that I took here at Salem State with Lynn Fletcher. And I thought, wouldn't it be nice to merge all these aspects of population genetics, phylogenetics, this sort of ecological sampling, and tick-borne disease, something that has some relevance to human health. So no one really explored moose for tick-borne diseases because they're really big and hard to catch. Anyone ever caught a moose? I did once. It didn't go well. It's like a Maine sleigh ride. It was in the water. So it ended up that I wanted to look at the big three, but getting into moose more, this is a keystone cultural species. If anybody's unfamiliar with that term, this is something that has an impact, a species that's going to have an impact beyond its biomass. Say, this is something that provides $70 million a year in hunting, tourism, whether it's ecotourism or something, if you're just staying at a hotel near Acadia. There's a lot of cultures up there that value moose at a much deeper level. I didn't realize that there was going to be so many Indigenous peoples talks at this event this year, but I think that's fantastic. Another aspect of my master's was working with the Penobscot and surveying them and looking at their perceived risk to the moose population versus what the actual risk is. So that was the grand overview. But I'm focusing more on the biophysical than the social science here because social science is really hard. the big three that I looked at, and this is not an exhaustive list of all the tick-borne diseases, but this is the ones you hear about the most. It's going to be Lyme disease, anaplasmosis, HGA in humans, and then a protozoan, Babesia. That's a eukaryote. The first two are going to be prokaryote. So what's interesting about all of these infections is you can see where they cluster. They're clustering very similar to where the moose were most populated. Another aspect about where moose are is, well, the moose are doing fine. There's tons of moose in Maine. But what keeps the moose under control are winter ticks. Winter ticks are a unique tick that has a 1-year, one-host life cycle. What makes deer ticks and dog ticks so effective at transmitting disease is that they go on a mouse, then they go on a deer, and then they might go on you. And each time, they're a walking syringe. They're going to be moving diseases that are harbored actually in the mice, propagated within the deer. They survive and they thrive through the deer, and then they might end up on you, transmitting the disease that doesn't live quite as rent-free in you. And that's where we have some human health relevance. All of these are very similar in the clinical symptoms when you look at anemia as a result of huge tick infestations on moose calves and look at the clinical effects of some of these diseases. Anemia, weight loss, all of these things are going to be basically indistinguishable on the surface. So if there was something that was harming these moose, what we've been looking at and studying for years is the effects of winter tick on moose calves. And winter tick is a species of Dermacentor. This is also the same genus as dog ticks. So they're larger than deer ticks, but because of that 1-year, one-host life cycle, they're not as great as transmitting disease or harboring disease, or so it was thought. So what I propose to do is do a complete screening. Look at the genetics, find genus-related primers to amplify in the blood anything that may be relevant or tissue for Borrelia species, that's relevant for Lyme disease, Anaplasma species, and Babesia. So what we would do is we collaborated. I was convincing, at least, with the person at UNH, and I got a parking ticket because I was late. But I managed to convince Pete Peekins at UNH to allow Inland Fisheries and Wildlife to collaborate with our new up-and-coming lab. And since it's been dubbed the Wildlife Disease Genetics Lab, with Pauline Kamenth, my advisor in that part of the work. So what we ended up doing was pretty straightforward. We were looking in a very new system for tick-borne disease. So we take our whole blood sample collection, we do DNA extraction. I know that's a QIAGEN kit. I'll get there. Look at really basic endpoint PCR, where we're going to take a little bit of DNA and amplify it to a large amount of DNA so that we can study it and visualize it. From there, we look at the sequence of those amplicons. This is going to be about 1,000 base pairs, the 16S rRNA sequence. This is a really nice region to look at for speciation because it's going to be a hypervariable but constant enough that you're going to look at minor changes. And all we're doing here is looking at how a laser will identify certain base pairs in the order in which they are sequenced in that gene. So we're able to compare all of these sequences, align them all, and based on the differences between those sequences, we can say, this is this % similar, and this is probably a different species or the same species. At a high level, that's what we're doing with phylogenetics. And then we can map them using models and make new discoveries and follow up. If I were doing this today, I would have used an NEB kit. They're better. They were cheaper back then, QIAGEN kits. Since then, this is a very long nested PCR that we were using. It would take 6 to 8 hours, depending on what classes you had that day. Since then, I've developed a lamp test. This is an isothermal test at NEB. This took about a day and a half. But this comparison of resources is unfair. What LAMP is really an exponential form of enrichment that happens all at one temperature. So these are the same types of things if you've ever taken a instant test or something like that. It's the same type of thing where everything's going to be contained, just like a qPCR, where you can get the extent of the infection, some sort of quantitative measurement. This is going to do something similar, but the way that it's different is it's not cycled with multiple temperatures, and it's also going to be using what it stands for is loop-mediated amplification, isothermal amplification. So that's what we developed. What a readout is just kind of a lot of these little pink and yellow buffers that you see at the end of your tubes. What this is it looks very clean. But this is a pH indicator. Every time a polymerase is adding a new nucleotide to your template, it's going to be releasing a proton that reduces the pH. As the solution gets a little bit more acidic, you start to see a color change. And this gives you a positive result in about 15 minutes instead of 8 hours. So this is how we're going to be doing all the testing in that lab moving forward. And this is the real results that we got from known positives and negatives. And it worked really well and even detected a sample we thought was negative. But we'll get to that. This is some gross pictures, because I can't not show you a gross picture of a calf dead, covered in ticks. Some of these moose calves in their first year can be found with mortalities with up to 70,000 ticks on them. What you see when you see a tick that's wicked fat and looks like this, that might be 8 milliliters worth of blood. That's a lot of blood. That's more than might fit in there because they're actually filtering the blood cells out and giving some of the liquid back. That's what makes them so great at transmitting disease. Anaplasma is going to propagate within the saliva glands, specifically anaplasma, but they're going to harbor in the red blood cells, sometimes the white blood cells for human infections, in different spots. But the common link is all of anaplasma, blood parasites, they're all going to reside in blood. So this is what it looks like for a little quadrant. We started getting a lot of positives with just the anaplasma, and then decided we should look under the microscope. Sure enough, we saw these little morulae. In humans, I think they're referred to as howl jolly bodies. Fun name. And this is particular to Anaplasma marginale, so named because it sort of exists towards the margins. There's a related species that exists towards the center that's called Anaplasma centrale. So it's easy to remember. Once we started getting results for Anaplasma positive and negatives, we started seeing an increase in anaplasma prevalence over the years, and that coincided increasing juvenile mortality. This was curious because we wanted to see if that increase in prevalence was contributing to the mortality, but it's hard to tease out. Remember I said that the anemia as a result of winter tick infestations would be really hard to tease out between subclinical infections of an anaplasma infection. This is, again, the life cycle of anaplasma. But when we mapped out the phylogenetics for anaplasma species that we were discovering in moose, this was one of those moments where you're kind of second-guessing yourself. You're kind of new to this part of the lab, and you're doing things 4 and 5 times just to make sure it's real. But I was clustering the anaplasma I found in the eastern moose in Maine with uncharacterized species that were found in Canada. So what that means, the short of it, is that we discovered a new species of anaplasma that was in moose. So you have now a new host, a new bacteria, and we're only in year 1. And it was a really cool find, and certainly gave me a jolt. So I ditched Lyme disease. I said, forget it, Lyme disease is really weird. And then Babesia, that one really wasn't working either. Anaplasma was the one that we started chasing. And this is a phylogenetic analysis that was run by me back then, and it's color-coded based on the anaplasma, but those icons are from the host that they were found in. So you can start to see a relationship. Where I have little tick icons, that's what I found in winter tick. I tested hundreds and hundreds and hundreds of winter tick, and because of that 1 year, one host life cycle that I mentioned, they're really not good at carrying bacteria. Turns out the literature was right. Every now and then I could find 1 or 2 Anaplasma phagocytophilum. That is what's relevant for humans. That's what's going to affect white blood cells. If the Anaplasma that seems to be in moose will not infect humans. But it was interesting that I couldn't find any of this uncharacterized Anaplasma species, which we dubbed Anaplasma cervus, because it's cervids, moose, deer. I wanted to call it Anaplasma Jimmy. They didn't want it, so that's fine. So we then started answering that question that I alluded to earlier. What is the difference between tick loads and Anaplasma infections? Came up with this really nice method of looking at survival over, I didn't come up with the method, I found a method, but started plotting all of our data over time, where the y-axis was survival probability, and the x-axis is time in weeks since birth. Since, conveniently, a lot of moose calves are born at about the same time. So the assumption was May 15th. So we moved on testing this. What I found is that when I looked at moose that had light tick loads, I saw no difference between Anaplasma positive and Anaplasma negative moose. So it could be very well that this is a subclinical infection that does not really harm the moose too bad on its own. When these are seriously infected, though, with a moderate or a heavy tick load, that Anaplasma infection really drives down the survivability of moose calves. This was very interesting because now we have the extent of the effect this bacteria is having on our moose population. And I want to be very clear, the moose population is doing just fine. This is classic predator-prey interaction that's going to self-regulate the moose population. They're not declining at an alarming rate. The actual risk is not that great. If I wasn't up here talking about winter ticks and Anaplasma, I'd be up here talking about how to get a moose off of everybody's driveway. So that's kind of a good thing, but it doesn't make it any easier to see calf mortality. Our objectives from there on, once we discovered that this system existed in our backyard, we started saying, well, let's estimate Anaplasma prevalence and distribution across their entire range. I left in 2019. We were very fortunate to acquire a lot of funding for a lot of students. One of them was going to be me. I left, I bailed, and everybody else that came in did a tremendous job expanding this beyond Maine. There's people, collaborators in Michigan, out in Washington, Alaska, up and through Canada now. And again, this started with just a little master's student, and it's really inspiring to see how wide this project has spread. Also, characterize the diversity of Anaplasma species infecting the moose. Is Anaplasma cervus, our new bacteria, the only thing that infects moose? Examine evolutionary relationships among moose Anaplasma strains and in the context of those found in other host humans and potential vectors. Primarily, I wanted to find the vector. Because of all the winter ticks I tested, I didn't find a single winter tick that had our bacteria of interest in moose. So where was it coming from? This is a really weird mystery. The new student that came in after me, Elena Woods, she is tremendous. She did a really great job connecting with hunters and started a program shipping kits that has everything that you might need to collect blood from their harvest that year. And she also got to interview a lot of these people, and she had great rapport with a lot of them, and the project would be nowhere near this big without her. She also expanded the wildlife management districts that they were studying in Maine. Back when I was working there, we had wildlife management districts 8 and 2. That was on the west there, and then at the very north. She expanded to almost the entirety of Maine, as well as outside of Maine. There were included a lot of moose captures that is done by helicopter, and it's the wildest thing you'll ever see in your life. These people are insane, jumping out of helicopters, wrangling moose. They don't like dart them or anything. It's just, they're not sedated. They're not killing moose. They're just doing a great job. They're all firefighters that you see diving into forests, and this is like their retirement gig, just catching big game. They're insane. So whole blood samples and then the ticks are both sent for anaplasma testing. This is the population sampling, kind of a map of what was done in New England and beyond in the Northeast, and as well as what we were able to test in the center of the U.S., of eastern moose population, and then an alternative subspecies that we were able to test out west. And so a really good sampling size of all of them. And so what anaplasma prevalence looked like by each state and province that we studied is that it was much bigger in the Northeast states, and out west it seemed to be on the rise, according to the data that we've collected so far. As we go a little bit into the colder regions, higher in latitude at least, we start to see less prevalence. What about the diversity? Anaplasma cerevisiae was by far the highest majority of the type of anaplasma that we found within moose. Oticolii and Phagocytophilum were also found. If you remember, Phagocytophilum is the one that is relevant for humans. But this is more prevalent as we go further out west. What is the story for this? Did Anaplasma cerevisiae evolve in the eastern part of the US and then start to spread west? Is this a sign of convergent evolution of the same species that's closely related to Anaplasma cerevisiae? Because there is if you remember the phylogenetic tree, there's a quite distinct drop-off. There's quite divergent, these two species, between Anaplasma Phagocytophilum, the one that's relevant for humans, and the one that we discovered in the moose. They're very divergent from each other. So the story is yet to be figured out in its entirety, but it's becoming very interesting. As we expand on the New England area, you can see that as we get a little bit colder, we move into the Anaplasma Phagocytophilum space. So there could be a climate-related story here. The updated phylogenetic tree is right here. This is the one that we worked on right before I left. And with some updated data involving the western part of the states. These two right here are, again, clustering with anaplasma that is relevant for human health. We also found anaplasma species uncharacterized in a totally divergent path within tabanin flies in Maine. These are biting flies that nobody likes. And then, most importantly, this was a fun story. It was the day before, I think 2 days before I graduated at Maine. I was in the lab cataloging all the ticks and making sure that the next person that comes in knows where all my stuff is. Because that would just be embarrassing if they came in and they couldn't find anything. So I'm going through all my tick samples, just kind of a disgusting task, ticks in jars, and moving through, and I found this one that did not look like a tick at all. It was a deer kid, after like an hour trying to identify it. It was this little insect that is also a biting insect that are found on moose. Where'd my head go? Is this my vector? I found that Anaplasma species residing in that deer kid. So now, in 2 years, we have a new bacterium in a new host that was not known to carry any tick-borne disease to any extent that was relevant to humans or to its own population, and a new vector, or at least a candidate vector, because this might not be where it's coming from. It might be just competent to receive it from the moose. There needs to be that distinction made. But this is a fun story for 2 years. Where we're at is that anaplasma prevalence in moose varies across the range, and it's also going to be varying in diversity. This is likely due to the climate, the species, and the vectors present in that area. Specifically, our bacterium that we have kind of fallen in love with, Anaplasma cervus, is a primary species infecting moose all across North America regardless. Deer ked, these insects that I just mentioned, are good candidate vectors, but more needs to be done in that system for sure. Moose are also infected with Anaplasma phagocytophilum. The extent to which they're contributing to human health needs to be investigated more. There were so many people that helped me and continue to carry on this project, and like I said, it's so humbling to see how far this has come. So I'm going to totally switch directions now to what I do on a day-to-day basis. I left University of Maine to come to New England Biolabs. And I wanted to tell a story rather than just talking about what I do on a day-to-day basis or teaching you about protein biochemistry and separations. I'll do a little bit of that, but it seemed a little bit more boring than I wanted to be. So NEB has been around for over 50 years, and it started with very humble beginnings in Beverly, looking at things like pregnancy testing, tRNA, and fell into this business of restriction enzymes. Before I go through the timeline briefly, how NEB came to be, our current campus is in Ipswich. This is our headquarters, but we spread all over the world now. Have subsidiaries in, I think, 9 or 10 different countries now. If you haven't visited our Ipswich campus, it is a lovely spot. I encourage you to take a drive-by across from Appleton Farms. How NEB came to be and what has led it to today is really the foundation was restriction enzymes. These are enzymes that are going to cut very specific sequences for very cut and paste like molecular biology that really founded the field for gene cloning. My first introduction to this, of course, was at Salem State with Tracy Ware. She taught me a lot about gene cloning, and it's all things that I still use today. When I'm asked, which classes do you still use in your day-to-day? Tracy taught 2 of them out of maybe 5. So shout out to Dr. Ware and all the work that she put into those courses. From restriction enzymes, the catalog has diversified quite a bit. We're now in RNA work, which I'm going to talk about more today. Amplification is a very big division now. Gene cloning and restriction enzymes are still a huge part of our business, but we're also moving into a lot of other aspects, including GMP grade manufacturing for facing regulated markets, and that happened just in time, right before COVID. And that's what I want to talk to today, to give context to the what do you do spiel. Operations and manufacturing is a little wacky, especially at a private company like NEB. It starts with the concept. Sometimes that comes from an application development department, sometimes comes from a commercial person within the company, and it really can come from wherever. Some ideas have come from within our group. I said, hey, it would be really nice during purification if we had XYZ, and then it becomes a product. That feels great. But in the process of operations, it starts with cloning, right? You're going to have some piece of DNA that's going to represent eventually the protein that you want to make, and you want to have that on some sort of switch, right? There's a lot of tricks to making, especially toxic proteins. But this is covered within the department called process development at New England Biolabs. That's where I reside. Cloning will work together with mass spec to make sure that we are making the thing that we're trying to make. And then upstream of me, people will learn and discover new ways for optimal expression. That is to say, as you grow things up in a fermentation tank, you're going to be culturing E. coli or whatever that might be as your host cell that's going to really be the workhorse manufacturing whatever enzyme that you want to sell. This can be as small as a 2 milliliter fermentation, or it can be as large as an 1,000 liter fermenter. The principles are the same. The engineering gets a little trickier though. After fermentation and cloning groups make a mess, it's my job to clean it up. When you bust open an E. coli cell, there's about 4,000 proteins. I want one of them. How do we get there? Well, if I were to summarize what exactly I do on a day-to-day basis, it's improve process understanding, quality, and efficiency for protein purification. That is isolating that 1 in 4,000 of those different proteins. Develop necessary high-throughput assays in order to do so. And then facilitate innovation. Go beyond what's off the shelf, right? Not to be limited by commercial and vendors and what they might be selling, but create your own technology to make that process development faster, more efficient. And of course, communicate your findings effectively. This helps no one if I keep it to myself. I discussed this workflow already, but I wanted to dive into what this type of separation means. How do we get one protein away from 4,000? And of course, all the other things that are inside that cell. Well, we do it with chromatography. This is the workhorse of what I do. A lot of companies sell these different resins that we can buy off the shelf that have different chemistries that will exploit different properties of your protein. A protein has unique attributes. And those attributes are what we exploit when we're separating proteins. That could be something by affinity. Something's going to be binding DNA. We can put something that looks like DNA on there. And everything that doesn't bind DNA will flow through that chemistry, and we can then elute, remove that one thing, fractionate it into different tubes, and then that's how we do all of this purification. We go through and tangentially exploit these different attributes. Through affinity, moving on from there might be something like electrostatic separations. Think positive, negative. These attract. Negative and negative repulse, positive and positive repulse. We can exploit the properties of our proteins and the environment to make these separations happen. There's also size exclusion. If I have a filter that excludes size of something, say, that's 100 kilodaltons, it's easier to separate from something that might be 30 kilodaltons. And then there's also hydrophobic interaction. This one's a little bit harder to understand, but it's the same principle as what you see when you mix oil and water. These are going to repel. These are just the different principles of separation. My readout on a day-to-day basis is something called a chromatogram. This can be done by gravity flow. You can have this sand-like material that is going to be have a sample with a mixed heterogeneous sample on it. All that sample will come through, and you start separating things by their interactions with that chemistry. My readout is going to be a variety of pumps, wavelengths that represent DNA and protein, conductivity, pH, all sorts of different readouts. And I use that to then analyze certain fractions. Using sort of these methods for protein detection, quantification, one of them being SDS page. This is a very high level overview. In this particular protein, it's very clear the most abundant of these proteins is the one that I'm after. But it's not as simple as that. There's also going to be tests to measure undesirable activity so that you can move away from the undesirable activity and towards the desirable activity. And you have to develop each of these assays for in-process testing for each new product. So in this case, I would look at the desirable activity, I'm going to take those fractions, and I'm going to move on to a new purification strategy until I have a single band and everything's at the quality I want it at. So who cares? The context for who cares, it used to be a lot harder to describe what I did before COVID. Now, I'm like, hey, you know that vaccine? mRNA? It needs certain elements in order to be non-immunogenic and to have high efficiency for functioning as a vaccine. As an introduction to what mRNA is, this crowd should probably know a lot of this by now, but it all is based on the central dogma of molecular biology, where we have a gene, turns into mRNA, mRNA encodes for protein. The process of going from DNA to RNA we know as transcription. So we started calling this in vitro transcription. In vitro means we take all the elements that you have in your cells, and E. coli might have in its cell, and we can take all these materials, optimize the enzymes that are required for these different steps, and put it in a tube. That's what the in vitro part means. New England Biolabs capitalized on a lot of the early companies that we collaborated with, like Moderna, when they were a younger company, who saw this vision of mRNA in vitro transcription being a scalable manufacturing process that could be used someday as a therapeutic. And everybody else knows the history, recent history, on how that came to be. When you look at this in vitro, in a tube, you're going to look at different aspects of the process. Really, we're going to simulate that gene component. We're going to generate a template to make our RNA to encode some sort of protein that will convey some sort of immunity in the context of a vaccine. Then we go through the transcription. This is done by an RNA polymerase. This is another enzyme. Excuse me. This is another enzyme that's going to be making RNA from a DNA template. But this was the problem with COVID. This was the bottleneck. Nobody's really been able to make a large amount of the enzymes that are responsible for maturing RNA to the point where it's functional. And this was for capping and tailing. Tailing is not so bad. It's using a poly-A polymerase. But Vaccinia capping enzyme was the legacy product for capping RNAs to make them functionalized. When I started in 2019, this is before the pandemic, I was assigned to work exclusively on capping enzyme. And so what capping enzyme will do is it'll make this structure called a CAP1 structure along with to achieving a CAP0 structure with another enzyme called CAP2O methyltransferase. But this is required again. If you do not have this, you can get an immunogenic response because it's recognized as a foreign agent in your body. It could look like, say, a virus. So the steps is we have something that was synthesized by an RNA polymerase from a DNA template. We end up with a triphosphate RNA. VCE comes in, puts a cap structure, which is the 7-methylguanosine material from a SAM methyl donor. And then we move on to our CAP2O methyltransferase, which matures this into a CAP1, sorry. And all we're doing from moving from a CAP0 to a CAP1 is right at the base of that mRNA where you have that 2' methyl group. So how do we achieve, I'd love to go into the details of how I optimized VCE in a process, but instead I'm going to have to talk about vague details because the benefit of working in New England Biolabs is that I can work on a lot of fun stuff, but I can't share a lot of it anymore. So what I'm going to do now is talk vaguely about different methods that we used to relieve these bottlenecks, increase the yields 100x, 1000x for VCE so that we could supply Moderna with all the reagents, one of our big customers, to vaccinate the world. And this was a really big task, and all of a sudden everybody wanted to look at my notebook, and it was a little bit intimidating. But we got through it, and it was a huge success story. So how do we go through it? Well, we operate under a different set of regulations than, say, pharmaceuticals. It's called research use only. Research use only does not have to answer to the FDA But that doesn't mean it's necessarily easy, because we're interested in activity and contaminating activities. Activities are much more sensitive than, say, the typical assays that are used to measure for impurities in pharmaceutical contexts. Because if the instrumentation in pharma cannot detect it, the correct assumption in most cases is that it won't affect our body. Research use only is very different. If we're making a polymerase, we don't want to have contaminating nuclease activity. There's no sense in making DNA if you have something in that system that's also chewing it. So that's what I was referring to between the desirable and the undesirable activity. So me and a few other colleagues in the process development group, which was just me and another person in 2019, and now is about 50 people, started looking at a bunch of different resins with a lot of different diversity within the chromatography space. All these different vendors from Japan to Sweden, also Biorad in California. We kind of surveyed 150 different resins to keep in our library. But what this means, once you start changing your proteins, your loading conditions, like pH and salt, and 150 different resins with different flow rates, different temperatures, you're looking at a very highly dimensional space. So you need high throughput screening. There is no computational analyses that'll tell you how to separate one protein that's brand new to science in many ways, in a lot of cases, from 4,000 other proteins. The parameters that I just mentioned that we can change in this workflow that we use for screening are the buffers, volume, flow rates, the loading titers, the concentration going on a particular volume. These are all customizable on a robot now. We set up all of the different things that we need so that all of the desirable and undesirable activities are measured as the output after whole purifications are conducted at a scale of 64 per run. This is something that we can gather in one day. How this actually looks like in real life, there's these little robocolumns that we can use to screen all these conditions, like I said, 64 in once. And they look like bullets. They get held up in customs a lot, because it looks like we're just buying bullets, but we're not. And then we use this robot that is our newest generation of automated system that we can then screen all of these different conditions. Our output looks something like this, where we can weight our variables based on our needs at that given part of the process, and then larger scale validation is done for every step of the process so that we can confirm that what looks good on a 200 microliter scale will also look good at a 40 liter column scale. And I guess the holy grail of bioseparations is customizable chemistry. We sell 750 products at New England Biolabs, depending on what you call a product. But there's a lot of enzymes, and there's 5 people in process development. And there's 40 products new a year. So we're busy, and we don't really have the luxury of having a lock and key fit for every single product that we make. Wouldn't that be great if I could design a new protein that only binds my protein, that 1 in 4,000, stick it to a bead, flow that whole heterogeneous complex over it, and just have my protein come out? Does it sound too good to be true? Yeah. It is. What's key is reversibility. You need to be able to get that protein off the column, and it needs to still have its function, because that's really what we're selling at the end of the day. We're selling a function. So designer ligands are too good to be true because they fail in some aspect of this. There's the reusability, robustness, specificity, stability, or cost. Some ways that people have succeeded in this are inteen-mediated splicing. This is something where an element in a protein you can clone in will use self-splicing methods that will actually remove a protein from a complex and mediate the ligation of 2 ends of a protein so that you can get around toxic effects, and also you can use this for affinity tag removal, that lock and key that I was talking about. One of the lock and keys that are used widely in a research use only space is called his tags. This is very simple. If you put histidines, an amino acid, at the end of an N terminus or a C terminus of a protein, it'll stick to metal. You can bump that off, it's reversible, it's a great step. But sometimes you can get metal toxicity. So again, sometimes too good to be true. What works really well are designer ligands, but they're very costly. And that's where that one falls short. Can't make designer ligands for 750 products. But what we can do is make possibly a designer ligand for its impurities. All of these are coming from E. coli or yeast or some other eukaryotic or prokaryotic host cell. If 90% of my proteins are made in the same host cell, I thought a few years ago, why don't we make designer ligands to remove the impurity? Because the impurity is constant. It's always the same nuclease. It's always the same things that show up. So that's what we did. We used something called aptamers. This is something that is going to be a single-stranded piece of DNA that forms some sort of 3D conformation that binds something. That is, it's very vague, but that is the definition of an aptamer. Has some sort of physiological effects. And in NEB, the aptamers are used widely, but not for this. We use them all the time for what's called hot starts. I can talk about that another time, but they're used for a different application. I started noticing that these were used widely in Taq polymerase, BST 2.0. This is something that we use for our LAMP assays, and it went on, and it was really gaining a lot of traction. We were developing a lot of new products with these aptamers. So how these aptamers, look, evolution, Darwin. How these aptamers are developed is systematic evolution of ligands by exponential enrichment. What we're doing is we're flowing a library of 10 to the 15 different sequences over something. Let's call it your impurity A. The things on that 10 to the 15 is a really big number. Inevitably, some of those are going to form complexes that bind your molecule of interest. We wash away unbound sequences, treat with proteinase K, amplify it, kinetically challenge it. That's a fancy word for dilution. And then we do it again. And we go through several rounds of this, and we end up then testing these at a much more thorough level using kinetics and various other things. In order to generate something that was an alternative to RNase inhibitor, something that's protein-based that we sell a lot of, a team at New England Biolabs patented, which is why I can talk about this, patented a technology looking at RNase-based aptamers. So this is something that if you don't want something to chew up an RNA, say like in an IVT workflow, you could put these aptamers in there. And even if there were contaminants, like RNase A or RNase I or whatever, this aptamer would block it. So it makes a contamination or an impurity event irrelevant. So it's a nice fail-safe to have if you're at a very large scale in a pharmaceutical setting. What I decided to do is I bet that would be really useful when I'm failing RNase assays for, say, my RNA polymerase. I took a bunch of enzymes out of the freezer, and I said, all these are failing. Let me take this aptamer that they just patented. I asked them to biotinylate it. I stuck it to a bead. Use these magnet beads to just treat my sample that was with a molecule of interest and an impurity. And I consistently, over dozens and dozens of samples, saw that I was removing that undesirable activity. So there might be something here. We started doing it with a bunch of other different nucleases, and we found a lot of success. So I just wanted to survey a little bit of each thing. Both of these techniques were used to alleviate that bottleneck that we saw with capping enzyme and a lot of other things that were challenges throughout the pandemic. I wanted to have this sort of unifying slide to end on, where so evolution connects us all. Understanding evolution dynamics will help us understand and respond to future challenges. We can predict disease emergence, something that was unifying through both of the topics that seemed very different today. Interpret our host-pathogen interactions, design robust, scalable biomanufacturing solutions like the aptamer-based technologies, and then from field ecology to process development, the same principles apply. That is variation, selection, and adaptability. I'll end there for discussion, and I'll take any questions.
Wonderful talk. Thank you so much. About the tick issue, what could you explain? What do you think the possible transmission vector is? So it's actually not the tick issue; it's the whatever that insect is.
Yeah. The deer head.
Yeah. And how do you test that? I mean, I know you aren't going to be able to do that, but how do you go forward trying to determine whether it's a vector? Because there are an awful lot of tick-borne things. I'm not aware of this insect.
Yeah, deer head are it's more of a wildlife issue. They're blood sucking and biting insects. Dr. Fletcher probably knows more about it. But the way that goes about being tested, that was the question: is what is the vector, and how do you test for a competent vector in a system like that? It's been a while since I thought about that, but there's a lot of literature looking mostly at Lyme disease. The way to Lyme disease, Borrelia, a spirochete bacteria, only is going to exist in connective tissue, so it's really hard to detect, right? So that's where those bacteria reside. And so when you talk about a competent vector, typically you start by culturing that bacterium in a plate with some sort of tissue. So you'll grow it in the connective tissue to study it, and then you'll do that so if the question is mice a competent vector.
Sorry to interrupt. Can you just talk to the microphone?
Yeah. So the question, yeah. If mice are the competent vector that's in question, you can take samples over time to see in a living animal if that bacteria is going up, and because the multiple things have to happen, right? The mouse can't get sick. It has to allow propagation of that bacteria, and then it also has to allow for another animal to take the anaplasma or whatever the bacteria is. So it's tough. I've seen it in cell culture where they study it in vitro, but I've also seen living organisms where it would be very difficult to keep a bunch of captive moose, for example. But people do it. Like there are wildlife sanctuaries in Maine that have captive moose, and I kind of even back when I was there, it was very young in the idea and the process, so it was kind of a hard sell to like, hey, can we just keep drawing blood from your moose? And it was also more of a problem with calves than adults, and that was an adult. So, but it's a good question, and it needs to be investigated more thoroughly because it might provide insight into how this bacterium has evolved here and maybe not out west, and how it continues to evolve. It would be interesting to overlay the distribution of deer head from here and out west to see if that competent vector sort of peters out, and that explains the difference in diversity. All sorts of ways. Thank you for the question.
So the question is, we're a Ketziologist.
Yeah.
So the tick that you looked at requires just one host, as you said, right, the winter tick. So all the species that you examined just require that. So what about the ones that are like transovarianly transmitted? Rickettsia parva has been shown in Canada as well as in moose populations, which has detrimental effects as well.
So the question was talking about vertical transmission and transovarian, and how generations of ticks can then propagate. That is possible. I don't know if it's possible with Anaplasma cerevisiae specifically within the winter tick, but Anaplasma phagocytophilum has been shown for sure to be transmitted through transovarian transmission. But there's also another way that we thought might contribute to how winter ticks are getting Anaplasma phagocytophilum in them is horizontal. So if they're feeding on the same animal that a deer tick is feeding on, that could be a way that they're transmitting it as well. So it may not be that there's a few different ways that we can get Anaplasma in the tick. It can be brought down by generation from mama tick, or it can be in an animal like a moose, and the winter tick is feeding on that moose, and then it's acquired maybe through an ixodes or another type of tick. Does that answer the question? Was that? I'm sorry.
That's fine. Yeah.
What was there? Was there another part? I'm sorry.
The actual question was, did you look at, so Anaplasma is not transmitted transovarianly, so did you look at other species that maybe were, since your model requires essentially just one feeding, all the species that really you examined are one feeders?
Yeah, I didn't look at any other ones, no. Sorry. Oh.
I just have one quick technical question. I don't know much about the lamp test that you were dealing with. I'm just wondering if there are gene expression applications of that lamp test, whether use it to look at different levels of expression of certain genes.
Yes, yeah, there are. I'm not an expert in that technology. I was able to develop it with the help of a friend of mine. And it was very straightforward with the primer design techniques that we have. But I know for sure that there's some like RT lamp that's targeted for gene expression, and it's very useful. I don't know how robust it is in vivo systems, but yeah, I can connect you with an expert for sure.
If I may have another question, it's about the capping.
Yeah.
So could you explain a little bit more about why that is so important for the mRNA vaccines, and that makes it look like it's a normal constituent of the cell?
Yeah. So as I was saying with the central dogma, for eukaryotes, all of our mRNA, if they're going to be translated from mRNA into protein, requires that 7-methyl-G cap and a poly-A tail. If it doesn't have that, my assumption is that it can look a little bit more like a virus or a bacterial expression system. Where it doesn't have that, it's going to be treated as a foreign particle. So a high titer, a high concentration of uncapped RNA does result in immunogenic effects. So undesirable symptoms, and that's not what we want in our vaccine. We want our vaccines to be preventing illness, not causing them, right? So that's why we need that cap. Other modifications beyond the cap, in humans, there always seems to be 2 base pairs in and adenosine that is always methylated, that makes it a little bit more attractive for effective translation. These cap modifications are kind of all the rage right now, and it's what I'm currently working on, and with a bunch of other stuff. NEB actively researching these cap modifiers to even greater increase the efficiency of some of these therapeutics. Hopefully that answers the question.
Yeah.
So it looks like we're about at the end. I just wanted to comment, Jimmy, and say it's totally amazing how much you did in 2 years. They should have given you a PhD for all that, I think. And to see like how it has grown, like after you left, and what you found, like what was it the day before you graduated, and then all that you've done at during the biolabs, like leading it. Anyways, I just hope you get the accolades you deserve, and I certainly recognize this amazing work you've done. Thank you for sharing with us.
Thank you so much.
Really great.